A massive volume of recent clinical research has isolated and quantified the specific health effects of fermentable dietary fibers. By definition, a prebiotic fiber survives the acidic environment of the human stomach. It bypasses our digestive enzymes completely, traveling intact into the lower intestine, where it serves as a primary food source for our gut microbiota. When gut bacteria consume these fibers, they initiate a fermentation process. This produces short-chain fatty acids, or SCFAs, which are vital metabolic byproducts that regulate inflammation and cellular energy. The clinical data reveals a precision problem with how we discuss nutrition. Fermentable fiber is a broad umbrella term, and structurally distinct fibers do not produce the same metabolic results. Take two heavily researched subjects, inulin and beta-glucan. Inulin is a storage carbohydrate found heavily in chicory root. Beta-glucan is a structural polysaccharide that forms the cell walls of oats and barley. Recent randomized controlled trials, or RCTs, which are strictly regulated scientific experiments used as the gold standard for clinical testing, along with comprehensive meta-analyses, show a clear biological divergence. Inulin and beta-glucan require completely separate clinical applications for microbiome modulation, glycemic control, and weight management. Advising a patient to simply increase their general fiber intake is an outdated clinical recommendation without specifying the exact prebiotic pathway. Inside the gut lumen, inulin acts on a highly specific set of bacterial targets. A 2021 meta-analysis confirms that inulin specifically targets and feeds bifidobacterium and lactobacillus. These are two distinct genera of beneficial lactic acid-producing bacteria, known to support gut barrier function. This selective proliferation is reliably triggered when patients consume 5 to 20 grams of inulin per day. Beta-glucan, however, drives a completely different microbial shift. RCT data demonstrates that beta-glucan selectively proliferates bacteria within the Bacteroides genus, a major group of microbes involved in breaking down complex carbohydrates. By feeding this specific genus, beta-glucan effectively expands the overall taxonomic diversity of the intestinal environment, improving general gut resilience. The foundational bacterial architecture of a patient's gut is directly engineered by the exact structural type of fiber they ingest. These microscopic bacterial shifts dictate exactly how the body controls systemic blood sugar. Inulin regulates glucose through a chemical and hormonal pathway. Short-chain fatty acids, produced by inulin, improve insulin sensitivity by unblocking cellular receptors, allowing the body to process glucose efficiently. In a 12-week RCT for type 2 diabetes, this hormonal repair caused a 0.3% reduction in HbA1c, the standard clinical metric for average blood sugar. A sustained drop of that magnitude signifies a meaningful, long-term improvement in fasting blood glucose levels. Beta-glucan operates through a mechanism that is entirely physical. When beta-glucan mixes with fluids in the gut, it forms a highly viscous, gel-like matrix. This gel creates a physical trap. It deliberately slows down the digestive process, physically delaying the speed at which carbohydrates are absorbed through the intestinal wall and into the bloodstream. This line chart shows beta-glucan's impact on post-meal glucose spikes. A meta-analysis confirms this physical barrier reduces post-meal sugar by 0.4 millimoles per liter. Contrasting the two, inulin creates long-term reduction in fasting glucose, while beta-glucan blunts immediate sugar spikes after a meal. Inulin repairs the body's hormonal response to sugar. Beta-glucan engineers a physical barrier against it. Both fibers successfully increase feelings of satiety, but their long-term clinical data regarding weight management diverges sharply. The data for inulin supplementation shows a high degree of friction. This scatter plot visualizes the clinical weight loss data associated with inulin. While patients report reduced meal intake, the actual long-term weight reduction is minimal. The data is highly susceptible to individual variance, and the statistical average settles very close to the baseline. The large-scale RCT data for beta-glucan tells a rigid statistical story. This bar graph tracks beta-glucan efficacy, highlighting a strict dose-dependent relationship for weight management. There is a precise intake threshold at 13 grams per day. Patients crossing this threshold effectively prevented weight gain and achieved an average weight reduction of 0.6 kilograms. Chemically induced satiety alone does not guarantee long-term weight loss. β-Glucan presents a statistically significant, dose-dependent mechanism for measurable reduction. Clinical outcomes rely on a tight therapeutic window. Health benefits are bound by highly specific dosing parameters. These dials display target intake zones. Optimal benefits require 5 to 20 grams daily for inulin and 3 to 8 for β-Glucan. Exceeding 30 grams of inulin generates excess gas, triggering severe gastrointestinal distress. This makes high-dose inulin a strict contraindication for patients with IBS. Inulin has a high ceiling before toxicity occurs, whereas β-Glucan requires tighter dosing precision. β-Glucan's strict daily dose maximizes at 8 grams for cardiovascular and glycemic optimization. Overconsumption pushes the digestive system too far, leading to mild but notable gastrointestinal discomfort. Extracting the metabolic benefits of prebiotics requires treating these fibers with the exact same dosing precision as pharmaceutical interventions. Zooming out from the individual trials gives us a holistic framework for clinical application. This synthesis matrix outlines the distinct clinical pathways side by side. Inulin relies on bifidobacterium to drive insulin optimization. β-Glucan utilizes bacterioids to engineer physical glucose blunting. Baseline microbiome makeup, existing metabolic states, and genetics create individual variants that will dictate real-world efficacy for any patient. For practical implementation, isolating these fibers via synthetic supplements offers exact dosing, but integrating them through whole food sources provides superior co-nutrient delivery. Current clinical consensus recommends sourcing β-Glucan from oat and barley formats, and inulin from chicory root, as the natural physical matrix of whole foods helps mitigate acute gastrointestinal side effects. True metabolic control via the microbiome is not achieved through a generalized increase in fiber. It requires targeted, dose-specific, and biologically appropriate prebiotic interventions.